So, good evening, students. Today we'll be discussing the most important concept of the brain, the blood-brain barrier. So, this is the one which protects the brain from all toxic substances entering the brain through the blood. The blood-brain barrier isolates the brain tissue from the rest of the body and is formed by the tight junctions that exist between the endothelial cells in the capillary beds. So we also have an additional barrier, blood CSF barrier, and we also have one more additional barrier, CSF brain barrier. The central nervous system requires a very stable environment in order to function normally. This stability is provided by isolating the nervous system from the blood by the existence of the so-called blood-brain barrier and the blood cerebrospinal fluid barrier. So, blood-brain barrier. The experiments of the Paul Ehrlich in 1882 showed that living animals injected intravascularly with vital dyes such as Trifonbut demonstrated staining of all the tissues of the body except the brain and spinal cord. So later it was demonstrated that although most of the brain is not stained after the intravenous injection of the Trifonbut, the following areas do in fact become stained. That means these areas are not having blood-brain barrier. That means the areas devoid of blood-brain barrier are pineal gland, posterior lobe of the pituitary, tuber cinereum, wall of the optic recess, and area postrema at the lower end of the fourth ventricle. These observations led to the concept of a blood-brain barrier. The permeability of the blood-brain barrier is inversely related to the size of the molecules and directly related to their lipid solubility. Gases and water pass readily through the barrier, whereas glucose and electrolytes pass more slowly. The barrier is almost impermeable to plasma proteins and other large organic molecules. Compounds with molecular weights of about 60,000 and higher remain within the blood circulatory system. This would explain why in the early experiments with Trifonbut, which quickly binds to the plasma protein albumin, the dye did not pass into the neural tissue in the greater part of the brain. So examination of an electron micrograph of the central nervous system shows that the lumen of a blood capillary is separated from the extracellular spaces around the neurons and neuroglia by the following structures. Namely, that means these are the three important structures which are forming the blood-brain barrier. The endothelial cells in the wall of the capillary. These endothelial cells are joined by the tight junctions and a continuous basement membrane surrounding the capillary outside the endothelial cells and the food processes of the astrocytes that adhere to the outer surface of the capillary wall. You can see here, this is the endothelial cell and this is an endothelial cell, and you can see this is the tight junction and this is the basement membrane, and these are the food processes of the astrocytes. Now, as we discussed earlier, there are certain areas where blood-brain barrier is not present, like pineal gland, posterior pituitary. So in those areas, so what is the structure of these capillaries? You can see clearly this is the endothelial cell, and here you can see the fenestrations of the endothelial cells. Fenestrations. So these are not actually direct openings, but there will be a small or thin, very thin diaphragm will be present, and you can see this is the basement membrane and this is the astrocyte. So that's why there is leakage of substances from here. So usually it is also clinically important because, like in hypothalamus, the hypothalamus will be testing the chemical concentration of the blood. The use of electron dense markers such as lanthanum and horseradish peroxidase has shown that these substances do not penetrate between the endothelial cells of the capillaries because of the presence of tight junctions that form belts around the cells. When the dense markers are introduced into the extracellular spaces of the neuropil, so which is a dense network of interwoven nerve fibers and their branches and synapses together with glial filaments. So neuropil, it is not neuropil A, it is neuropil. Neuropil is nothing but it's a dense network of interwoven nerve fibers and their branches and synapses together with glial filaments. So when the dense markers are introduced into the extracellular space of neuropil, they pass between the perivascular foot process of the astrocytes as far as the endothelial lining of the capillary. That means what they are trying to say is, even from outside, they are trying to enter into the capillary. Only the tight junctions between the endothelial cells is preventing and acting as a barrier. On the basis of this evidence, it is now known that the tight junctions between the endothelial cells of the blood capillaries are responsible for the blood-brain barrier. In molecular terms, the blood-brain barrier is thus a continuous lipid bilayer that encircles the endothelial cells and isolates the brain tissue from the blood. This explains how lipophilic molecules can readily diffuse through the barrier, whereas hydrophilic molecules are excluded. Although the blood-brain barrier exists in the newborn, there is evidence that it is more permeable to certain substances than it is in adult. The structure of the blood-brain barrier is not identical in all regions of the central nervous system. In fact, in those areas where the blood-brain barrier appears to be absent, the capillary endothelium contains fenestrations across which proteins and small organic molecules may pass from the blood to the nervous tissue. So it has been suggested that areas such as the area posterior of the floor of the fourth ventricle and the hypothalamus may serve as sites at which neuronal receptors may sample the chemical content of the plasma directly. The hypothalamus, which is involved in the regulation of the metabolic activity of the body, might bring about appropriate modifications of the activity, thereby protecting the nervous tissue. So next one is the blood cerebrospinal fluid barrier. There is free passage of water, gases, and lipid-soluble substances from blood to the cerebrospinal fluid. Macromolecules such as proteins and most hexoses other than glucose are unable to enter the cerebrospinal fluid. It has been suggested that a barrier similar to the blood-brain barrier exists in the choroid plexus. So electron microscopic examination of a villus of a choroid plexus shows that the lumen of a blood capillary is separated from the lumen of the ventricle by the following structures, namely. So earlier in blood-brain barrier we have seen only the three layers were there: endothelial cells with tight junction, basement membrane, and the foot process of astrocytes. But here we are seeing five layers, namely the endothelial cells, which are fenestrated and have very thin walls, then continuous basement membrane surrounding the capillary outside the endothelial cells, and scattered pale cells with flattened process, continuous basement membrane on which the choroidal epithelial cells will rest. The use of electron dense markers has not been entirely successful in localizing the barriers precisely. Oxidative peroxidase injected intravenously appear as a coating on the luminal surface of the endothelial cells and in many areas examined it did pass between the endothelial cells. It is probable that the tight junctions between the choroidal epithelial cells serve as the barrier. You can see here the blood cerebrospinal fluid barrier. So this is the area or the lumen of the capillary. You can see these are the endothelium, and this is the basement membrane of endothelium. These are the process of pale cells, and this is the endothelium. This is the basement membrane of the choroidal epithelial cells, and these are the choroidal epithelial cells. And you can see here this is the tight junction between the choroidal epithelial cell. So last but not least, cerebrospinal fluid brain interface. Although vital dyes given by intravenous injection do not gain access to most brain tissues, if the dyes are injected into the subarachnoid space or into the ventricles, they soon enter the extracellular spaces around the neurons and glial cells. Thus, there is no comparable physiological barrier between the cerebrospinal fluid and the extracellular compartment of the central nervous system. It is interesting, however, to consider the structures that separate the cerebrospinal fluid from the nervous tissue. Through three sites may be examined: the pia covered surface of the brain and spinal cord, the pia covered surface of the brain and spinal cord, the perivascular extensions of the subarachnoid space into the nervous tissue, and the ependymal surface of the ventricles. You can see here outer surface of the brain. Outer surface of the brain, you can see these are the foot process of astrocytes. This is the basement membrane, and you can see here these are the cells of the pia mater. These are the cells of the pia mater, and this is the connective tissue network in the subarachnoid space. This is the arachnoid matter, and this is the dura mater. So if you see from outside inwards, arachnoid matter, this is the dura mater, arachnoid matter, then this is the pia mater, and this is the basement membrane, and these are the foot process of astrocytes. Then the other one is from the inside, ventricular surface of the brain. You can see here these are the choroidal epithelial cells, otherwise called ependymal cells. Ependymal cells which line the ventricles. These are the ependymal cells which are lining the ventricles, and you can see this is the tight junction between the ependymal cells, and these are the process of astrocytes and other glial cells. So these are the two surfaces which have been examined for cerebrospinal fluid and brain barrier. So the pia covered surface of the brain consists of loosely arranged layer of pial cells resting on basement membrane. So beneath the basement membrane are the astrocytic foot processes. No intercellular junctions exist between adjacent pial cells or between adjacent astrocytes. Therefore, the extracellular space of the nervous tissue are in almost direct continuity with the subarachnoid space. The prolongation of the subarachnoid space into the central nervous tissue quickly ends below the surface of the brain, where the fusion of the outer covering of the blood vessels with the pial covering of the nervous tissue occurs. The ventricular surface of the brain is covered with columnar ependymal cells with localized tight junctions. Intercellular channels exist that permit free communication between the ventricular cavity and the extracellular neuronal space. The ependyma does not have a basement membrane, and there are no specialized astrocytic foot process because the neuroglial cells are loosely arranged. So what is the functional significance of the blood-brain and blood-cerebrospinal fluid barriers? So in normal conditions, the blood-brain and blood-cerebrospinal fluid barriers are two important semi-permeable barriers that protect the brain and spinal cord from potentially harmful substances while permitting gases and nutrients to enter the nervous tissue. So last, to add a functional significance with the all these blood supply, we'll just have few points regarding the cerebral circulation. So what is actually happening? The blood flow to the brain must deliver oxygen, yes, definitely, then glucose, definitely, this is the main energy source, and then other nutrients to the nervous tissue, and at the same time it has to remove carbon dioxide, lactic acid, and other metabolic byproducts. So the brain has been shown to be supplied with arterial blood from two internal carotid and two vertebral arteries. So the blood supply to half of the brain is provided by the internal carotid and vertebral arteries on that side, and their respective streams come together in the posterior communicating artery at a point where the pressure of the two is equal and they do not mix. So that means internal carotid and vertebral systems are not normally mixing. If, however, the internal carotid or vertebral artery is occluded, the blood passes forward or backward across the point to compensate for the reduction in blood flow. This is the most important point why the circle of Willis has been formed at the base of the brain. The arterial circle also permits the blood to flow across the midline as shown when the internal carotid or vertebral on one side is occluded. It also has been shown that two streams of blood from the vertebral arteries remain separate and on the same side of the lumen of the basilar artery and do not mix. Although the cerebral arteries anastomose with one another at the circle of Willis and by means of branches on the surface of the cerebral hemispheres, once they enter the brain substance, no further anastomosis occurs. The most important factor in forcing the blood through the brain is the arterial blood pressure. This is opposed by such factors as a raised intracranial pressure, increased blood viscosity, and narrowing of the vascular diameter. Cerebral blood flow remains remarkably constant despite changes in the general blood pressure. The autoregulation of the circulation is accomplished by a compensatory lowering of the cerebral vascular resistance when the arterial pressure is decreased and a rising of the vascular resistance when the arterial pressure is increased. Needless to say, this autoregulation does not maintain an adequate blood flow when the arterial blood pressure falls to a very low level. So the diameter of the cerebral blood vessels is the main factor contributing to the cerebrovascular resistance. While it is known that cerebral blood vessels are innervated by sympathetic postganglionic nerve fibers and respond to norepinephrine, they apparently play little or no part in the control of cerebrovascular resistance in normal human beings. The most powerful vasodilator influence on cerebral blood vessels is an increase in carbon dioxide or hydrogen ion concentration. The reduction in oxygen concentration also causes vasodilatation. It has been shown by using positron emission tomography that an increase in neuronal activity in different parts of the brain causes a local increase in blood flow. For example, viewing an object will increase the oxygen and glucose consumption in the visual cortex of the occipital lobes. This results in an increase in the local concentration of carbon dioxide and hydrogen ions and brings about a local increase in blood flow. The cerebral blood flow in patients can be measured by the intracarotid injection or inhalation of radioactive krypton or xenon. A cerebral blood flow of 50 to 60 ml per 100 grams of brain per minute is considered normal. Thank you. So most important thing, just see this slide. 30 years from now, it won't matter what shoes you wore, how your hair looked, or the jeans you bought. So what will matter is what you learned and how you used it for the benefit of humanity. Thank you.